DNA, the special code inside almost every cell in your body that makes up who you are. Whether you are one of 20 quadrillion minuscule ants, or a colossal whale that weighs hundreds of tons, DNA is the main component of every living organism. But what if we could change it? Be as tall as a skyscraper, have the muscle mass of an elite bodybuilder, even glow like a jellyfish in the deep blue sea. Like, the possibilities are endless.
Alright, maybe I'm getting too ahead of myself.
Am I though? Because recently, scientists have been making traits that wouldn't normally happen. They've gained the ability to modify DNA in ways that we wouldn't really thought possible, such as giving animals extra limbs, changing their colors, and even splicing DNA.
Even though this is fascinating, this makes you wonder, what relation does this have with evolution? Forcing organisms to change this rapidly? Could this lead to repercussions? All of this eventually led to my final question. How long does it take for evolution to happen, and does genetic modification play a role? Or does it do something far more sinister?
Now, before I did anything, I needed a plan. A plan that would answer my question, a plan that would be cheap and accessible, whilst also being high quality and efficient. And after a lot of research, I had found the holy grail of all plans. I decided I was going to bioengineer something myself and see if I could spot any of the differences between bioengineering and evolution, whilst looking for the effects it may have on the organisms. And this is how I did it. Now, bacteria, like all living things, use DNA as instructions to make proteins. Proteins are tiny machines that do everything from helping us digest food to making our muscles work. In this experiment, I gave non-pathogenic, which means does not contain or produce disease, E. coli bacteria a special gene from a jellyfish. This gene makes a glowing protein called GFP, green fluorescent protein. And here's how I did it. Now, before I begin the step-by-step process, I just want to clarify some things. It is impervious to follow proper protocol when doing any science experiment. I'm lucky in the sense that Odin, the creators of the kit I used to modify the bacteria with, provides a document with all methods and steps required. Thank you for listening so far, and let us begin. Step 1. I prepared the bacteria. I mixed freeze-dried bacteria with a special solution to make them competent, which means ready to take in new DNA. Step 2. I added the jellyfish gene. I combined the bacteria with a circular piece of DNA, called a plasmid, that carries the GFP gene. Step 3. Heat shock. I briefly warmed the bacteria to help the DNA get inside their cells. Step 4. Let them grow. I placed the bacteria on plates with agar, food for bacteria, and let them grow for a day. At the end, I used blue light to see the glowing jellyfish bacteria, which means the DNA transformation worked. This experiment shows that evolution, a process that normally takes millions of years, can be dramatically sped up with bioengineering. Traditional evolution relies on random mutations and natural selection over generations, but genetic engineering allows us to make precise changes of DNA in just days or even hours. By inserting a glowing jellyfish gene into bacteria, we gave them a completely new trait almost instantly, something that would have never happened naturally. While this demonstrates the power of bioengineering to solve problems or create new possibilities, it also raises ethical concerns. If we can rewrite the genetic code so easily, it's important to consider how this technology is used, whether for good, like curing diseases, or in ways that could have unintended or harmful consequences. Thank you for watching my presentation. Have an amazing day.